Hello everyone. In this presentation, we will be discussing immunosuppressants. So as the name indicates, immunosuppressants are drugs that have the capability of reducing the activation and the efficacy of the immune system. So the question is, why would you want to suppress the activity of the immune system? We all know the importance of having good, efficient immunity because that is the primary mode of how you fight infections, damage by foreign objects. In fact, there are several reasons why can we use immunosuppressants in clinical medicine. The main use for immunosuppressants is for the treatment of autoimmune diseases. Remember that autoimmune diseases are special medical conditions where the immune system abnormally recognizes the normal tissues and cells in the body as foreign objects. So the immune system will start attacking normal organs and tissues in the body and damage them. This abnormal activation of the immune system results in several diseases, we call them autoimmune diseases, such as systemic lupus erythematosus, rheumatoid arthritis, and inflammatory bowel diseases, and so on. There are several diseases that fall into this category. So the solution is to use immunosuppressants to lower the immune response and thereby decrease the damage caused by the immune system. In addition, for the treatment of autoimmune diseases, immunosuppressants can also facilitate organ transplantation. Remember that with organ transplantation, a foreign organ is harvested from a donor and is implanted in the recipient's body. So this new organ is considered as a foreign body, which will elicit an immune response. And for that, if we want to achieve a successful organ transplantation and prevent the body from rejecting the newly transplanted organ, we will have to suppress the immune system. That's where we also use immunosuppressants. So the search for those methods to successfully exert organ transplantation are very old. People tried to use organs from deceased individuals and transplant them into patients who are in need. This thought uh, was realized by humans early on during history. There were multiple failed attempts to transplant skin grafts, for example, to repair burned skin or injured skin. Some people also thought about transplanting eyes. However, the first proper kidney transplant operation was performed in 1933, and that failed because of immune rejection. The kidney uh, transplanted into the patient was rejected by the immune system. At the time, we poorly understood the function and the role of the immune system, and we had no immunosuppressant drugs available. And it took us 20 years from 1933 to be able to successfully do the first kidney transplant operation. And that was definitely through the help of newly developed drugs such as 6-mercaptopurine and azathioprine. Both of these were the first immunosuppressant drugs to be used in organ transplantation. From that point and on, more drugs have been developed, and the technology of organ transplant has improved. And that led not only to kidney transplant, but to more advanced transplant operation of other organs, such as the pancreas. The early drugs that have been used to facilitate organ transplantation had several adverse effects. With time, the drugs have been improved and that was mainly when cyclosporin was introduced into clinical medicine in the 1980s. Cyclosporin became one of the fundamental immunosuppressant drugs used for organ transplantation. We then were able to perform the first successful cardiopulmonary transplant in 1981. After that point, more and more drugs have been developed for that purpose. Now, note that while immunosuppressants are used for the treatment of autoimmune diseases, and some of them are also used for the treatment of certain malignancies, as we will discuss shortly, the focus of this lecture will be on how these drugs are implemented and used for organ transplant. So, as I mentioned, the aerial drugs that were used as immunosuppressants, such as azathioprine and 6-mercaptopurine, these drugs were non-selective, they were cytotoxic, they were basically chemotherapeutic agents similar to those used for the treatment of cancer, and they suppressed the immune system on both the humoral and cell-mediated immunity. Remember that we have two types, or more than two types of immunity. We have innate immunity, humoral or antibody-mediated immunity, and cell-mediated immunity, which is mainly mediated by T cells. So these drugs were non-selective and would affect all um, components of the immune system, which led to many adverse effects. Now, the recent drugs, the newly developed drugs, can selectively target certain proteins, certain processes associated with lymphocyte activation. So these drugs had more activity, they were more effective, and they were associated with less adverse effects. So in order to understand how immunosuppressants work, we need to just quickly review how the immune system, especially lymphocytes, recognize foreign antigens. So let's say, for example, that a bacterium has entered the body. Okay, this is a pathogenic bacterium that will be recognized and processed after its entrance to the body. And this is mainly engulfed by phagocytic cells such as macrophages. So macrophages will engulf the bacterium. So the bacterium is now inside the cell. And macrophages, as well as other types of immune cells called dendritic cells, both of these are termed antigen-presenting cells, which means that these cells will metabolize the ingested foreign body, in this case the bacteria, and will process the antigens of the bacteria, mainly bacterial proteins, and then present those antigens on its surface. Okay, so macrophages and dendritic cells will present those foreign unique antigens of the bacteria on their surface. Now, these antigens can be recognized by lymphocytes, mainly by T cells. So T cells will be able to use their receptors. There are different types of receptors that can recognize these antigens. Once the T cell recognizes the antigens presented on macrophages and dendritic cells, these T cells become activated and they can then activate other types of lymphocytes, such as B cells, that are responsible for antibody production. So in this case, B cells are the cells that are highly involved in humoral immunity, or the immunity that involves the production of antibodies that help the body to recognize these foreign antigens in case that pathogen has entered the body again. This means that the activation of T lymphocytes is a very critical step in developing an immune response. The activation of a T lymphocyte happens through a very complex process that involves three main pathways. Now, note that it is not enough for a T cell to just only see the abnormal or the foreign antigen presented on ABC. This step is just only the first step in T cell activation. So in that step, T cells using their T cell receptors bind the foreign antigen presented on macrophages and dendritic cells. In addition to that, there should be another signal we call the co-stimulation signal, where the T cell interacts directly with the antigen presenting cell using receptor-receptor interaction. In that process, CD80 and CD86 receptors found on macrophages and dendritic cells will bind CD28 receptors found on T cells. This binding will enhance the activation of T cells. The last step is cytokine mediated, meaning that T cells become activated by the binding of different interleukins, such as interleukin 2, which binds to IL-2 receptors located on the surface of T cells, resulting in their proliferation. So let's discuss those three steps in more detail. As I mentioned, the first signal that is required to activate T cells is the binding of the T cell receptor, or CD3, to the antigen presented on the antigen presenting cells, such as macrophages or dendritic cells. So this binding activates the T cell receptor that results in elevated intracellular calcium levels. So that binding increases calcium inside the cells. Now, calcium is definitely a very important signaling molecule, and rapid increases in calcium levels will activate a protein called calcineurin. Calcineurin is a calcium-dependent protein phosphatase, and as the name indicates, calcineurin is activated by high levels of calcium. So when calcineurin is activated, it can dephosphorylate. Remember, it's a phosphatase enzyme, which means it takes out phosphate groups from molecules. Calcineurin can then convert another protein called NFAT, or nuclear factor of activated T cells, by removing the phosphate group attached to that molecule. This converts NFAT from its inactive form to its active form, which means it can then travel from the cytoplasm to the nucleus, where it can exert different functions. Mainly, it activates the transcription of many genes that would lead to the uh, uh, production of messenger RNAs of several cytokines, including IL-2 or interleukin 2. This will facilitate the synthesis of many inflammatory and immune mediators. The second step in the activation of T cells involves the binding, the direct binding between the CD28 receptor located at the T cell surface with CD80 or CD86 receptors located on the antigen presenting cells. So this is a direct receptor-receptor binding. This elicits a co-stimulation signal that even activates the T cell furthermore. The last step is when IL-2 binds to its receptor present on the surface of T cells. Of course, this can happen in an autocrine fashion, meaning that the IL-2 produced by the T cell can go back and then stimulate the same cell, or it can stimulate other cells that also have the receptor. Binding of IL-2 to its receptor on the surface of T cells results in a signaling pathway that eventually activates a very important signaling molecule called mTOR, or the mammalian target of rapamycin. mTOR is involved in many processes inside the cells. One of those is that it drives cells through proliferation, right? So it results in rapid cycling in the cell cycle and consequently more cellular growth and proliferation. So the last step mediated by IL-2 results in the growth and proliferation of the activated T cells. You need all these three steps in order to obtain an effective activation of the immune system. So now, based on that, immunosuppressants are used for two main purposes in terms of organ transplant. Some of these drugs are used for the induction of immunosuppression, meaning that they will result in a rapid suppression of the immune system in order to be able to implant the new organ. And these are mostly monoclonal or polyclonal antibodies and are very potent immunosuppressants. On the other hand, there are a separate set of drugs that are used for the maintenance, meaning that after the organ has been successfully transplanted, you will need to lower the immune drive for a prolonged period of times to avoid rejection later on after the surgery. And for that, we use a different set of immunosuppressants that are mostly not as potent as the drugs used for induction, and they are associated with less adverse effects. So for the purposes of this presentation, we will start discussing the drugs that are used for the maintenance immunosuppressive therapy. We have four main categories of drugs that are used as maintenance immunosuppressant agents. The first category are called the calcineurin inhibitors. Both drugs that belong to this category are called cyclosporin and tacrolimus. So let's take a look and see how do these drugs suppress the immune system. Remember what we just described, that as part of signal one of activating T cells, increased calcium levels will result in the activation of calcineurin, which then activates several signaling molecules, basically the in-fat transcription factor that results in the activation of transcription of many inflammatory genes. This means if we inhibit calcineurin function, we will be able to interfere with signal 1 and prevent the activation of T cells. Cyclosporin binds to an intracellular protein called cyclophilin, while tacrolimus binds to another intracellular protein called FK binding protein, or FKBP. Binding of both drugs to their respective proteins results in the formation of drug-protein complexes. These drug-protein complexes are highly active and they can directly inhibit calcineurin function. And that's how these drugs prevent the activation of T cells by interfering with signal 1. Because of their potent immunosuppressant effects, both cyclosporin and tacrolimus are used for the maintenance of immunosuppression during organ transplant. However, tacrolimus has now become the preferred calcineurin inhibitor and the mainstay of treatment, especially when applied for solid organ transplantation, such as kidney or heart transplant. Remember that we can also do soft tissue transplantation, such as bone marrow transplant. And the reason for that is because tacrolimus is associated with a better safe or safer profile. Of course, these two drugs, because they are immune suppressants, they will definitely increase the risk of infections. This is basically the most important problem associated with most immunosuppressants, and that's why patients who take these drugs, who have undergone organ transplant, will be susceptible for bacterial and viral infections. Cyclosporin can also result in direct kidney damage or nephrotoxicity, and that was one of the reasons why it was replaced by tacrolimus. The second category of maintenance immunosuppressants are drugs that interfere with the co-stimulation step 2 in T cell activation. Remember that signal 2 involves the direct binding of CD8, CD86 receptors on the antigen presenting cells to the CD8, CD28 receptor on the T cells. So the drug belatacept, which is a co-stimulation inhibitor, is a recombinant fusion protein that binds CD8, 80 and CD86. So it binds to the receptors located at the antigen presenting cells. When it binds those receptors, it blocks them and it prevents them from binding to the CD28 receptor on T cells, meaning that belatacept interferes with this step 2 or signal 2 of T cell activation. Belatacept is highly used for a maintenance therapy in kidney transplantation, usually in combination with other immunosuppressants. Note here that the majority of immunosuppressants are used in combination. And the reason behind that is that these drugs have several adverse effects profiles. They can be highly toxic. So we use them in combination at lower doses in order to lower their toxicity. In addition, we use several drugs of several mechanisms of action to produce effective immunosuppression. This is very similar to the concept of how we use combinational anti-cancer chemotherapy in the treatment of cancer. Belatacept can also be used as a substitute to tacrolimus to avoid long-term nephrotoxicity. Remember that both cyclosporin and tacrolimus can be associated with kidney damage. Belatacept seems to have a safer profile than both drugs. However, there are still some limitations with using belatacept. This drug should not be used in patients who have been infected or have an active infection with Epstein-Barr virus, because belatacept can result in a significant suppression of the immune system, allowing for the activation of Epstein-Barr virus, resulting in a unique condition called post-transplant lymphoproliferative disorder or disease. This serious condition can happen after using belatacept as a maintenance immunosuppressant therapy after transplantation procedures. The third category of maintenance immunosuppressants are mTOR inhibitors, and drugs that fall into this category is the drug sirolimus. Remember that the third step or third signal required to activate the proliferation and growth of activated T lymphocytes involves the binding of IL-2 to its receptor on T cells, which results in the activation of mTOR signaling. So the drug sirolimus or another name for that is rapamycin. Remember that mTOR is named after the drug. mTOR stands for mammalian target of rapamycin. So this is the protein that is the target for this drug, sirolimus or rapamycin. So rapamycin is a potent inhibitor of mTOR, and it blocks its downstream signaling pathway, leading to cell proliferation, meaning that sirolimus and everolimus block the proliferation and growth of activated T cells, and that also leads to suppression of the immune system. Note here that the inhibition of mTOR is not direct. Sirolimus and everolimus, both these drugs need to bind with the FK binding proteins in order to form a drug protein complex, and that drug protein complex then is capable of inhibiting mTOR function, meaning that it's very important that FKBP or FK binding proteins to be available in the cell in order for these drugs to exert their effects. As I mentioned, inhibition of mTOR will result in inhibiting several translation processes, such as the synthesis of many cytokines involved in immune system activation, and will result in T cell growth arrest, mainly in the G1 phase of the cell cycle. It will prevent further progression through the cell cycle. The last category in maintenance immunosuppressants are drugs that directly interfere with the proliferation of T cells. These drugs are basically cytotoxic therapy, and they are very similar to the cytotoxic anti-cancer chemotherapy. Both drugs fall into this category are azathioprine and mycophenolate. Both of these drugs were the first drugs to be developed as immunosuppressants. So azathioprine directly interferes with lymphocyte proliferation by blocking or inhibiting nucleic acid synthesis. Remember that cells of the immune system, such as lymphocytes, are rapidly growing cells. They're daily generated from the bone marrow. They're rapidly dividing. And these cells require or are highly dependent on nucleic acid synthesis because they undergo several cell divisions. They need to synthesize their DNA. So azathioprine, being a prodrug, is first converted to a metabolite called 6-mercaptopurine, which is also an immunosuppressant drug and a known anti-cancer drug as well. And mercaptopurine is then metabolized into thioinosinic acid, which is a nucleoside analog. Okay, it has a structure that is very similar to nucleosides. This means that this analog, as well as 6-mercaptopurine, they both can be incorporated into the growing DNA chain. So let's say that the cell is synthesizing its DNA. Its DNA polymerase is building the daughter DNA strand. We want to replicate the DNA and form another DNA molecule. During that process, if 6-mercaptopurine or thioinosinic acids were present, they will be converted into nucleosides, and they will also be mistakenly inserted into the newly synthesized DNA, because DNA polymerase will think that they are normal nucleosides. They have very similar structures. So when these analogs are inserted into the newly synthesized DNA, they will block further elongation of DNA. Okay, they will act as brakes, and that will extensively inhibit DNA synthesis, and thus will inhibit the proliferation of T cells. On the other hand, mycophenolate blocks effective lymphocyte proliferation by interfering with the de novo synthesis of nucleotides. That means that it will deprive the cells or will reduce the amount of nucleotides available for DNA synthesis. So mycophenolate is a potent, reversible, non-competitive inhibitor of the enzyme inosine monophosphate dehydrogenase. This enzyme is responsible for the conversion of inosine monophosphate into xanthine monophosphate. Xanthine monophosphate is then converted into GMP or guanosine monophosphate. And remember that guanosine monophosphates are a major component of the newly synthesized DNA, right? You need GMP, you need CMP, AMP, and TMP to synthesize DNA, right? G, C, A, and T. So this pathway leads to the synthesis of GMP. And mycophenolate, by inhibiting the enzyme IMP dehydrogenase, it blocks the formation of GMP, reducing the availability of these nucleotides for DNA synthesis, which rapidly blocks the proliferation of T and B cells. Both azathioprine and mycophenolate are used as adjunctive immunosuppressants in addition to calcineurin inhibitors. Remember, as we discussed previously, that these drugs were the first to be used. They were used alone, but because of their non-selective profile, because remember, these drugs just interfere with the proliferation of any growing proliferating cells. They're not specific to T cells. They can inhibit both T cells and B cells, as well as the function of other rapidly growing immune cells. That's why they are associated with high risk of infections. So these drugs are used in addition or as adjunctive or as complementary drugs to calcineurin inhibitors. Mycophenolate has largely replaced azathioprine because it has a better safety and efficacy profile. Now let's discuss corticosteroids. I understand that these were not included in any of the four previously discussed drug classes, but corticosteroids were amongst the first and most effective drugs to be used for suppressing the immune system. They're all drugs, and we know that corticosteroids can lower immunity, and they can also have very potent anti-inflammatory effects. That's why corticosteroids are used for the treatment of autoimmune diseases and chronic inflammatory conditions because they suppress immunity and they suppress inflammation. That is basically the main use of corticosteroids. They're mainly used against autoimmune diseases. However, they are still considered one of the mainstay treatments used for immunosuppression in cases of organ transplantation. The most common form of corticosteroids used is the drug prednisone and the drug methylprednisolone. Both prednisone and methylprednisolone are drugs that have multiple uses, and you will be learning about corticosteroids in more details when you take the endocrine system. Unfortunately, we don't exactly understand the mechanism of action of corticosteroids and how they effectively suppress the immune system. We definitely know that they have both anti-inflammatory effects and immunosuppressive effects. We also know that T lymphocytes are the major component of the immune system that is affected by corticosteroids, as corticosteroids can directly kill lymphocytes and reduce their population. In addition to that, we know that corticosteroids enter the cell and bind to the steroid receptors that are located in the cytoplasm. This activates steroid receptors, which then translocate into the nucleus, where they mediate the production of many cytokines and vasoactive substances, including IL-1 and IL-2 and IL-6. And remember, all of these interleukins are very important for the activation of the immune system. So in this graph below to the right, you can see that corticosteroids can exert many effects that lead to immunosuppression, including reducing the number and the function of lymphocytes. They can cause lymphocytopenia or reduced number of lymphocytes. They can also lower complement levels, and they can interfere with the synthesis of many inflammatory cytokines, such as the interleukin families that lead to lowered leukocyte function. So corticosteroids have wide uses. They can definitely be used for both the induction and the maintenance of immunosuppression for organ transplantation. They can also used for to suppress acute rejection of solid organs allografts, like if a kidney was transplanted to a recipient, and shortly after the recipient started developing an acute immune response against the transplanted organ. We call it the rejection process. Corticosteroids can immediately be given, mostly intravenously, to suppress immunity and facilitate the acceptance of the organ. Again, the most common use of corticosteroids is for the suppression of the immune system in autoimmune diseases. That's why we use them to treat SLE or systemic lupus erythematosus, rheumatoid arthritis, temporal arthritis, and asthma, and so on and so forth. They have wide medical applications, and they're very important drugs. However, corticosteroids are not very selective, and they are associated with many adverse effects. Remember that these are derivatives of endogenous cortisol. It's another hormone released by the adrenal glands, and it's involved in the metabolism. It's not just, doesn't only affect the immune system. This is just one part, but endogenous corticosteroids are involved in many functions of the body. Um, and that's why corticosteroid therapy can result in a diabetogenic effect, meaning that corticosteroids result in hyperglycemia, they increase glucose levels in the blood. They can also increase the levels of cholesterol in the blood, and both of these effects are associated with increased risk for heart disease. Patients who are on corticosteroid therapy will gain weight rapidly, and you probably have seen that before. Someone who's taking cortisone, for example, for the treatment of something, and they start gaining weight very rapidly. They can also cause some eye disorders. And because cortisol results in reduced calcium deposition in bones, these drugs can cause osteoporosis if they were used for a prolonged period of time. So understanding the pathophysiology of corticosteroids explains the high profile of adverse effects associated with their use. So now let's move and discuss drugs that are used for the induction of immunosuppression. These are the drugs that we use in order to facilitate the implantation of the organ before the surgery is performed. Most of these drugs are either monoclonal or polyclonal antibodies. Okay, we have two types of antibodies. These drugs are more specific and more selective. Polyclonal antibodies are generated by injecting human lymphoid cells into animals, into horses and rabbits. So when you inject these human lymphoid cells containing antigens of interest, the immune system of the horse or rabbit will generate a mixture of different antibodies directed against that antigen. These antibodies, which circulate in the plasma of those animals, can then be collected and harvested. Now, we call them polyclonal, is that because they originate from different immune cells. So let's say, for example, you have a foreign body produced by a bacteria or something. So this foreign body will be recognized by different immune cells from different angles. This is just for simplicity. This means that cell A will produce an antibody against that antigen that is different from the antibody produced by cell B. Okay? So despite that these two antibodies are directed against the same antigen, they're not totally the same. They are derived from two different clones of immune cells, and that's why we call them polyclonal. These are usually less selective. Monoclonal antibodies, on the other hand, these are derived from one clone of immune cells, from only one cell, and they are directed more selectively against an antigen. So the way that these antibodies are generated is using the hybridoma technology. This involves that you inject the antigen of choice that you want to generate those antibody against into the animals, usually mice. Then the mouse will respond with uh, 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 will activate its immune system against that foreign antigen, and its immune system will produce antibodies. Remember that antibodies are majorly produced by plasma cells. Plasma cells are the main antibody producing cells in the body. So you can then harvest those plasma cells and choose the clone of interest. So one clone of plasma cells producing one antibody on its surface. This plasma cell can be then fused with a cancer cell. The reason why we do that is that you want to fuse the genetic material located in plasma cells responsible for the production of the antibody of interest with the highly proliferating cells, such as the cancer cell. So the cancer cell will continue growing very fastly because that's what cancer cells do. And that means that you will increase the production of the antibody of interest because now this genetic material is inserted into the tumor cell. That's why you're creating a hybrid cell between plasma cells producing the antibody and the tumor cell that is growing very fastly. And that allows you to generate uh, uh, large amounts of the monoclonal antibody of interest. Of course, we can genetically edit. Now, remember, these plasma cells are derived from the mouse, meaning that these antibodies are mouse antibodies. So we can also humanize them, is that when the hybridoma is made, we can edit the genes responsible for antibody production and make them more similar to human DNA. And that process is called humanization of antibodies, and it's usually associated with less antigenicity and less adverse effects of these drugs. So to quickly compare both types of antibodies, polyclonal antibodies are cheaper to produce. You only need to inject animals with the antigen of choice, but they are associated with a mixed population of antibodies. That's why they're less specific, and uh, they have effects that are different from monoclonal antibodies, which are in turn are expensive to produce. However, they are more specific and they find the antigen of choice more specifically. So there is a way of how we name these drugs. Note that the suffix MAB or MAB is usually used to denote monoclonal antibodies. For example, drugs such as basiliximab idarucizumab. These drugs, because they have mab at their ends, this means that these drugs are monoclonal antibodies. Now we can use other references as well. The letters XI means that this antibody, this monoclonal antibody, is chimeric, meaning that it's derived from animals, from the mouse. Or they can have the two letters Z and U, which means that this drug was developed by a hybridoma involving gene editing, resulting in a humanized antibody. So, for example, idarucizumab is a monoclonal antibody because it has mAb at the end, and because it has ZU as well, it is considered a humanized monoclonal antibody. On the other hand, basiliximab is a monoclonal antibody, but it's chimeric, meaning it's a mouse human antibody. So let's start talking about basiliximab. As I just mentioned, basiliximab is a chimeric monoclonal antibody, of course, produced by the hybridoma technology. It results in a prevention in T cell proliferation. And the way it does that is that basiliximab directly block the binding of IL-2 to its receptor on the surface of T cells. Thus, it interferes with signal 3 of T cell activation, which leads to prevention of mTOR activation and inhibition of T cell proliferation. So the effect of basiliximab results in T cell growth arrest or inhibition of proliferation, but not exactly killing T cells, okay? Not depletion. C cells will remain, but they won't grow as effectively. Basiliximab is used to prevent acute rejection. It's an induction drug, especially in transplantation of kidneys. And if it was used properly, it's highly effective that can delay the use of maintenance therapy, such as calcineurin inhibitors. Okay, it was very strong from the beginning, and that will allow you to delay the use of calcineurin inhibitors. Rituximab, on the other hand, is also another chimeric monoclonal antibody. However, rituximab does not target T cells. In fact, its target is a receptor called CD20, and CD20 is a receptor located only on B cells, mainly pre-B cells, mature B cells, and memory B cells. Okay, all of these cell types express the receptor CD20. Binding of rituximab to the CD20 receptor will result in killing of B cells or B cell lysis. It will definitely lower the numbers of B cells significantly. It will also interfere with B cell activation and prevent the maturation of B cells into antibody-producing plasma cells. Remember that mature B cells then proliferate into plasma cells, and these are the cells responsible for the production of antibodies. This means that rituximab directly interferes with humoral immunity or antibody-mediated immunity rather than interfering with the cell-mediated immunity that is usually mediated by T cells. So this drug kills B lymphocytes. Remember also that it only works against CD20 expressing cells, such as I said, pre-B cells and mature B cells and memory B cells, okay? Uh, Plasma cells do not express CD20, okay? This means that rituximab does not kill plasma cells. And if at the moment that rituximab is given, there will be some plasma cells circulating in the plasma that they won't be killed by rituximab. Rituximab, because of its ability to interfere with humoral immunity, can be used for organ transplantation, for the induction for sure, especially to facilitate implantation and to reduce organ rejection. But because of its ability to target B cells, it can be used for the treatment of autoimmune diseases such as rheumatoid arthritis. It can be used for the treatment of certain cancers, such as B cell lymphoma, right? Because B cell lymphoma and certain types of leukemia, these originate from B cells. And those tumor cells also express the protein CD20. This means they can be targeted by rituximab and killed by rituximab. Lastly, rituximab can be associated with a life-threatening rare viral infection caused by the reactivation of JC virus. So this virus is an opportunistic microorganism, meaning that is under normal immunity conditions, it is inactive in case the patient was infected. But once rituximab is given and it lowers the immune drive, JC virus can lead to a life-threatening disease called progressive multifocal leukoencephalopathy. So that's why rituximab has a black box warning in that it increases the risk of infection. It is associated with hypersensitivity reaction, and it can cause the fatal, life-threatening disease called progressive multifocal leukoencephalopathy. Bortezomib is a proteasome inhibitor. Okay, this drug results in inducing apoptosis or cell killing of plasma cells. Remember that rituximab has no effect on plasma cells, right? Because they don't express CD20. However, bortezomib can kill plasma cells directly. Thus, it is another example of a drug that targets humoral immunity only, immunity mediated by B cells and plasma cells, not the immunity that is mediated by T cells. It is again used to suppress antibody-mediated rejection in transplant patients. It's used for induction, but it also can be used to treat certain types of blood cancer, such as multiple myeloma. Remember that multiple myeloma is originates from plasma cells. Okay, thus the tumor cells in multiple myeloma express they are susceptible to bortezomib. Okay, bortezomib selectively targets plasma cells, so we can also use it to target tumor cells derived from plasma cells in multiple myeloma. Okay, now we come to another concept called intravenous immunoglobulin. Intravenous immunoglobulin is just a general term that describes several types of immunoglobulins that circulate in human plasma. We can isolate those immunoglobulins from the blood donated by humans, right? Because we have blood banks full of blood samples. Some of those samples will be used to harvest the immunoglobulins or the antibodies already found in those blood samples. And we call those intravenous immunoglobulins because we can give them to other patients through the intravenous route. This drug is often referred to as IVIG. And despite that, we don't understand what's the mechanism of action of these wide heterogeneous immunoglobulins isolated from human blood. They can cause immunomodulation and they can directly result in B cell apoptosis. That's why we can use them for the treatment of autoimmune diseases or to induce immunosuppression organ transplantation, or to reduce antibody-mediated rejection as well. So many uses. They are associated with several adverse effects, including headache, fever, chills, hypotension, or hypertension. Of course, IVIGs are also considered in many other conditions, such as platelet disorders, and they are used, for example, against idiopathic thrombocytopenic peptides, which is a bleeding disorder common in children.